Hi, my name is Mark Jacino. I'm a field application specialist with Buki Corporation. Today I'd like to discuss some features on our flash chromatography system that highlight ease of use and help decrease the amount of time normally spent on sample purification. These are the topics we'll cover: system configurations, software, detection, Navigator software, and we'll talk a little bit about some natural products applications and the use of up to 4 solvents in one run. What is flash chromatography? It's a technique used to clean up and concentrate samples. It's a means to isolate and collect material for additional testing. Always performed prior to quantification steps such as HPLC, GC, MS, and so on. Uses inexpensive disposable cartridges. It's a fast technique and uses the same method development principles as HPLC. The system that we have to do flash chromatography is our Reveleris X2. Here's some of the feature sets on this instrumentation that we're going to cover today. It has a nice large size touchscreen. The unit is very compact, as you can see by the height, width, and depth measurements. Easily fits in the hood or fits on a laboratory desktop. Does not take much space. Has automatic cartridge detection, has pressure sensors, vapor sensors in case there's a leak, automatic injection valve rinsing, anti-static solvent lines, selective detector sensitivity for low and high concentrations of samples, uses a high pressure HPLC pump, has auto priming features, and it comes with multiple detection systems. The preparative purification system that we offer is based on the flash system, and it includes the flash system. This particular unit, you can run flash, or you can go to a high resolution mode and run on a preparative system. As you notice, the same size instrument, same height, width, depth, same software as you can see, same high pressure HPLC pumps, except this unit will run at higher pressures when using the automatic injection valve for preparative chromatography, allowing you to use standard preparatory metal columns. This instrument comes with a very intuitive software interface, very easy to use, very quick to learn. As you can see on this screen here, this is more or less the setup screen and the screen that you'll be seeing the entire time you're working with the instrument, unless you go to a tools menu where you're going to be able to do something additional, such as an injection port rinse, solvent line flush, naming a different solvent of your own creation, etc. So this is the view that you'll always be working with. The controls are easy. The system is set up easily for you to find and set your conditions very quickly. There are adjustable flows and pressures during the run. You can change a lot of parameters during the run, such as the gradient profile, adding additional solvents, increasing the run length, changing the flow rate, changing the threshold sensitivities. As I mentioned, the same software is used on the prep and on the flash system. So if you notice here, these are the screens from both units. The main difference is on the prep system. If you go to the mode selector, you can switch from preparative chromatography to flash chromatography. So there's no other hardware connections or no other things to do. If you have the flash column in place and you have the prep column in place, all you really need to do is switch from one to the other to be able to use either side. You'll notice on the flash mode screen, there's flash at the bottom at the center and prep at the bottom at the center to let you know which screen, or which mode I should say, you're in. What's the idea behind having automated flash? and having multiple detection systems. The process of purification, crude sample, reaction workup, sample pre-screen, flash separation, all the way down to submitting for testing, all these particular steps take time. The more time they take, the less other particular projects can be worked on. So by being able to free up time at any of these particular steps increases productivity. The main places where we're going to see is here with the flash system that we offer is saving time with less samples to be tested and easy to set up, less peaks to collect, and thus, once again, less samples to be tested. And then at the end, once you're submitting, you don't have to worry that samples or contaminants that may be in the system may not have been detected by your specific detector set. Fraction collection. This is what Flash is based upon. You're triggering a collection to collect a compound in a pure state. Most systems offer a UV triggered collection. Very common, very good way of collecting fractions. Most HPLC systems run on this concept for analytical work. However, we also find that in some cases, there may be non-chromophoric compounds. There may be solvents that interfere because they have UV activity. And because of this, we could end up missing particular samples. As you can see on the right side of this panel, UV detectors sometimes can't see things such as lipids, waxes, salts, sugars. Sometimes a contaminant is not visible to the MS either. Most cases, what we do find though is all potential non-volatile compounds that might be in your sample can be seen by the ELSD. What is ELSD? Evaporative light scattering detection is a system where the sample is nebulized, so it's formed into a series of very small droplets. These droplets are evaporated, so the solvent shell around the particle is evaporated off, and then these particles pass through a light source, in case of our particular instrumentation, a laser, and this causes those particles to scatter light, which is collected by a photodiode. Why employ these types of detectors while doing your purifications? Well, the main thing here is that an integrated ELSD maximizes recovery of your compounds. It can detect compounds with less volatility than the mobile phase. It detects non-chromophoric compounds, targets contaminants, salts, sugars, provides better qualitative sample information. Because of this, it presents a better component ratio than UV. A strong or a weak absorber in UV will have a subsequent strong or weak peak. On the ELSD, the peak is always going to be based on the amount of particles, so the higher the concentration of material, the larger the peak response would be. This also lets you determine relative mass balance, so you can see what type of concentrations you have. Enables the use of chromophoric solvents. If acetone or toluene or a strong absorber, even if you have to run your assay at 210, then you want to use methanol. All of these will be an issue if you're using a UV-only system, not an issue at all with an ELSD. This also minimizes the need to collect all fractions. If between the UV and the ELSD, you more or less have a universal system where you should be able to detect anything. So if there's no response, you shouldn't have to worry about testing those particular fractions. This causes post-analysis workup to be minimized. You don't have to TLC every test tube and collect every drop of the run. And this, at the end, provides more confidence in subsequent testing results and decreases time spent on testing results. Minimizing fraction workup. So here's an example of this. So now we are only going to have to check peak fractions that we see, because we know that all targets and impurities have been identified. Fewer number of fractions at the end of the assay reduces your volume to work with. It also reduces the number of TLC plates. It also reduces the number of post-collection assays that need to be performed for quantification. On the top panel, we have a system with ELSD and UV. We collect peaks only, and we notice that we have 3 peaks to analyze at the end of the run. On the bottom, we're doing a UV only, so we have to collect all of these fractions because we only see 1 peak. However, if we didn't collect all 14 fractions in this particular case and analyze them, we would miss peak 1 and peak 2. Sample purification. Should we use flash, prep, or both? This is a manual gravity chromatography column. Almost everyone's familiar with this type of work. While it does have high capacity and high sample loading, it offers poor resolution, very slow sample purification. While there are multiple phases available, more or less whatever material you want to buy in a drum and pack in there, it does take a lot of time, and there's no direct detection. So all the samples and fractions that are collected must be tested independently. If we move over to automated flash chromatography, we find we have high capacity, we have very good resolution, we have very fast sample purification. We use inexpensive and disposable cartridges. Multiple phases are available on different cartridges. There's multiple detections that can be used, either UV or ELSD in this case. And the results are very reproducible from run to run. Finally, for people that need extra resolution, the automated prep style chromatography can do 2 things. It can run in the prep mode, or you can run the flash mode. Samples that are purified by flash can be polished on the prep side to yield even higher purities. So on this side, we're looking at lower capacity on the prep side only, because we're using HPLC style prep columns. We have very high resolution because of that, very fast sample purifications. However, sometimes the prep columns can be a little expensive, but any phase out there available can be used with this system from any manufacturer. Once again, there's multiple detectors available for UV and ELSD, and these results are highly reproducible. Flash versus prep. So what does that mean when we look at those 2 instruments I just talked about? High resolution, high purity, available on both, and why would you want both? Well, here's an example of a crude extract. We have a compound of interest that we can see here at the top panel. HPLC run was done to get an idea of the separation we'd be looking at. Now, if we look at that information that we gained and set up a run, we'll find that on the flash, which is high capacity, this particular case, I was able to load 10 grams of material onto the cartridge. We find our compound of interest, but it's not highly resolved. Now, if we look at the prep assay, which was done with about 1 gram of material, we find a very, very similar pattern on the prep column that we saw on the HPLC column, which are both running as high pressure columns. It's a little bit easier to delineate your particular peak of interest on the prep side from a specific HPLC run. However, when we find the analysis for purity by LCMS, we find that both of these yielded very, very good results. The prep side is a little cleaner because we had a little less material coming over from some of the other little bit cleaner fraction cut, because the separation was baseline to baseline. However, the material collected from the flash run is very clean and would need to just have a polishing run to bring it up to the same purity level as the prep run. And we were able to generate 10 to 1 when it came to the amount of material collected back. If we look at an example here of a prep purification, this is also looking at different modes here. So we have purification technique where we're looking at citral from lemongrass oil. So we're looking at purity numbers and recovery. If we look at the flash system, we're looking at about 98%, and we're looking at about 360 milligrams per gram recovered. On the prep system, we're looking at 99% and about 388 milligrams per gram recovered. And on competitor style prep system, we find that we're looking at about 96% and about 373 milligrams per gram recovered. So what we see here is that the flash and the prep system are both very efficient at doing this, as efficient as many of the standard, what we think of as a standard style preparative system, just a HPLC style system that can be purchased from many suppliers out there. However, the prep and the flash prep are much more economically priced and are much more flexible because you can do a lot more material and then polish it and get the same purity levels. Let's talk a little bit more about some of these productivity steps and some more highlights on this. Here's a case study where we looked at a synthesis, a standard synthesis step. So something that anyone in a pharmaceutical firm or in a med chem style situation would be doing. They're taking some molecular components, they're reacting them, and then they're coming up with a synthetic molecule that they've created. And in many cases, there's some leftover reactants, some side products, things like that that need to be purified out. If we follow the flash purification process, and we look at this little chart here, we can see a standard UV system versus the flash with ELSD system. Now we're looking at, as we looked at before at the beginning of this discussion of all the process steps, the reaction workup, sample pre-screening, flash separation, sample workup again, and then what's the total time frames for these. So what we're looking at here are more or less minutes involved in doing these steps. And we find that in almost every case, by using the flash system and the ELSD, allowing us only to have to look at specific collected peaks and not at a large volume of collect all style samples where we're not quite sure if there's material in these or not, we find that we see about a 50% time savings, which is a significant improvement for productivity. Purifying to achieve optimal results. The flash system that we have, our Reveleris, comes with a Navigator software feature that allows for rapid method development. Method development can be a time-consuming process. In many cases, in flash purifications, a scout run is done first. After that's collected and tested to see if it's what the purity is, then a secondary run with more material can be done with more confidence so that you'll know that the purity level will be high enough because the material has been separated well enough. But if purifications can be improved for quality and speed, these will decrease costs. So the Navigator feature will allow you to input initial HPLC or TLC conditions. It will optimize the gradient starting conditions. It can even warn you if the separation is not predicted. If there's not enough separation, then the instrument, or the Navigator feature in this case, will warn you that a different solvent set is probably preferred and will ask you to rerun the TLC plate using a different solvent set. And we will also find that we can use multiple solvents to increase resolution and extraction, especially for people doing natural products. Let's look at a quick example again. This is a triazole reaction. So same thing, we're doing a synthetic step. We're looking at product that's formed. And then we also have some unreacted material left over, which needs to be purified. So our compound of interest needs to be purified. And these other trace materials, unreacted materials, need to be removed. So if we look at using the software for this particular instance, We're going to input our RF values, input our solvents that we're using. We're going to input the type of cartridge we want to use, the size, the flow rate, and then we're going to decide if we want to continue to 100% to flush the cartridge out at the end of the run. And we're going to also decide if we want to run this assay for speed or purity. Speed assays decrease the amount of time and solvent used, but still maintain a good alpha, so you have enough resolution to purify the product. Purity will increase the runtime by using a shallower gradient, and this will allow for even larger alpha between the peaks, and therefore a much better or a much higher purity number, especially if there are a little bit more closely related peaks. These are the gradients generated from our particular data set from the TLC plates. So we have purity run and we have a speed run. We'll notice on the purity run that the gradient starts lower. We start at a higher percent A, and we also have a shallower gradient. On the speed one that we're looking at, we can also see that on both of these, actually, we can see that it's been checked to continue to 100%. So what we see is there's a hold added to the end of the assay to allow the column to be flushed out with 100% of the B solvent. These are our results for purity and for speed. So what we can see here on the purity run, we're starting at 0% B, and we have a shallow gradient. And what we're looking at, although the run is a little bit longer, our samples are coming out at about 4 minutes. They're beginning to come out to about 6 minutes. We find that we have a nice resolution for our peaks. If we look at the speed run, now we find our materials coming out at about 1 minute, and it's done by 3 minutes. We're starting at a little bit higher percentage, and we have an adequate resolution to still look at our separation. However, in this case, after 4 minutes, the run is effectively over, and the solvent percentage could be automatically raised to 100% by the user during the run because all of those steps are editable. Here's our analysis of our product. So we find that they're absent of the starting materials or contaminants. Both speed and purity methods work very well. And so we look at a final product purity of near 100%, and we have a very quick, rapid separation that was done by simply doing quick set of TLC plates. The instrument generated a suggested separation paradigm, automatically inputted into the system, so you don't have to worry about changing any settings. It'll automatically take those settings and push them over onto the run screen. Simply make sure the cartridge is in place, inject your sample after the equilibration, and run your sample and collect your purified material. There's another example that we'll look at. In this case, we're going to do a little bit different. We're looking at, once again, a synthetic chemical reaction to produce a specific target compound. We have some reactants, some other materials left over, plus our compound of interest after our reaction. In this case, what we're going to do is look at purifying this by using the Navigator once again, but in this case, instead of using TLC plates, we're going to use the LC transfer function. So what we've done here is we've taken a crude sample, analyzed it by an HPLC method, and then what we've done is we've transferred the information from the HPLC method into the flash system by using the retention time data, and this will give us a predicted paradigm for separating the material on the flash system. So a very small amount of our sample can be used to generate the method on the HPLC system. If we see that we have adequate resolution on the HPLC system, then we can transfer that to the flash system and now run a much larger scale, so a much larger capacity run. Instead of micrograms, now we could be looking at multi-gram separations. Hi, my name is Mark Ticino. I'm a field application specialist with Buki Corporation. Today I would like to discuss some features on our flash chromatography system that help highlight the ease of use and how it helps to decrease the amount of time spent normally on sample purification. These are some of the points that we'll cover today: system configurations, user-friendly software, detection, Navigator software to increase productivity, and then we'll look at some other extractions and purifications at the end that can use up to 4 solvents. What is flash chromatography? Flash chromatography is a technique used to take a complex mixture and separate it into its component parts. It's used as a means to collect these for additional testing, so these purified compounds can be elucidated. It's done prior to any quantification step using HPLC, GC, MS, and so on. Flash chromatography uses disposable, inexpensive cartridges, and it utilizes the same method development principles as HPLC. Here we see an example of our Reveleris X2 system. You can see from the dimensions that this is a compact system, fits easily inside of a hood or on a laboratory benchtop. Up to 4 solvent bottles can be put on top. Solvent bottles aren't really any higher than the touchscreen that you see. So once again, these are easily managed in a hood. They're not too tall for the hood to be. Hi, my name is Mark Ticino, and I'm a field application specialist with Buki Corporation. Today I would like to discuss some features on our flash chromatography system that highlight ease of use and help decrease the amount of time normally spent on sample purification. These are some of the points that we'll go over in today's discussion: system configurations, system software, system detections, Navigator software, and we'll look at some extractions and purifications using these particular detectors and software. What is flash chromatography? Flash chromatography is a technique used to clean up and concentrate complex samples. These isolated materials can then undergo additional testing, such as HPLC, GC, MS, for purity or for compound elucidation. This technique uses inexpensive disposable cartridges and operates under the same principles as HPLC, where a method is developed and then your separation is performed. Here we see the X2 flash chromatography system. You can see from the dimensions that this is a compact system, does not require a lot of hood space, or if it's on a laboratory bench top, not a lot of bench space. Solvent bottles can be put on top. They don't extend past the touch screen, so these fit easily in a hood and can be managed in a hood. System comes with several safety features such as anti-static solvent lines, comes with cartridge pressure sensors, vapor sensors in case there's a leak or a spill, has an automatic injection valve rinse. It uses RFID tagged trays, so it knows the size of the trays that are in the system so it doesn't overfill them. It also has sensors on the solvent bottle lines and on the waste line so that the waste can't overfill or the solvent bottles can't run dry during a run. Here we see our prep purification system, which is a modified flash system. We see it's the same dimensions, we see the same safety features, we see the same sizes, we see the same software on the screen. In this case, what we're really looking at, if we look at the front of the instrument, we're looking at an automatic injection valve for preparative chromatography to be run on the system. Both of these systems run with high pressure HPLC style pumps. So running preparative style columns, preparative metal columns on this type of system is a good way to get very high resolution on complex samples. Let's take a look at the software that's used on these systems. It's very intuitive, easy to use software. Everything is really on this one screen. This run setup screen here allows you to pick your solvents, load your solvents, pick your detectors, pick your sensitivities, pick your thresholds, set up your gradient profile, set your collection parameters for how you want to collect your samples, the amount of sample to be collected, even tells you the amount of solvent that you'll use while you're running this assay. Here we see the same screen on both the flash and prep system. Both systems are identical in their appearance, other than the prep system having the injection valve on the exterior part. On the interior part, there's some different plumbing that allows it to run at a much higher pressure, but both use HPLC style pumps, so they both have very, very good gradient control and very, very good solvent delivery. It's very easy to switch between the flash and the prep system on the Reveleris flash, or the Reveleris prep system. In the mode drop-down menu, you simply pick which side of the instrument, the flash side or the prep side, you'd like to use. And if you look here on the bottom, you see the flash screen and the prep screen. The only difference is that one says it's in flash and one says it's in prep. On the flash system, you're always going to be the same screen that you see on the flash mode system. Purification process. The purification process can be a time-consuming and difficult process depending on the compounds that are being purified. Detecting more compounds from your crude mixture not only reveals more information about the sample, but can also free up more time because you don't have to worry about missing particular components that might be there. All of these steps, making the crude sample, reaction workup, pre-screening it, the flash separation, checking the collected fraction samples, all of these, and the subsequent testing, all of these particular steps take time. Anytime you can save some time on any of these steps means that there's more time that can be focused on other projects or other work. So if there's less workup to be done, that'll save time. If there's less TLC visualization methods that need to be done, if the sample pre-screening and the flash separation can be set up rapidly, if the collection peaks versus collecting all peaks, so you only have a few peaks to sample or a few peaks to test for purity and for identification, then all the material that's run through the system. And then knowing that the peaks that you did collect represent everything in the sample, and then you don't have to worry about particular materials showing up later after the purification process. How are we going to visualize these samples to trigger the collections? Most HPLC systems and most flash systems use what's known as a UV triggered purification. So as the material moves through a flow cell, it absorbs UV light that generates a signal. Once the signal passes a threshold, that material is collected. Very common method, very time-proven method. However, it can only collect things that are chromophoric. It can only collect things well that are at the right wavelength also. So what can happen is non-chromophoric impurities or targets of interest can be missed. UV active solvents can interfere. Acetone is a solvent of choice, or you're running at a low UV absorbance like 210, or you're using a very active solvent like toluene. All these things can make UV triggered collection much more difficult. However, if you look on the right, we notice that if we look at a UV set of detection, MS detection, and a light scattering detection at the bottom, we notice that the ELSD seems to see everything. The reason behind this is the ELSD can see any type of particle, chromophoric or non-chromophoric, as long as it's a non-volatile item. So salts, lipids, sugars, as well as chromophoric items such as possible compounds of interest for pharmaceutical companies. All of these things can be easily seen with the ELSD, and by coupling that with the UV detector, which can see volatile components, we can then have a universal system where we can more or less see anything that's coming out of the instrumentation. How does an ELSD work? Why would I want to have this ELSD on the system? The ELSD is somewhat of a unique method of detection because it's looking at either the absence or presence of something. So if something has a mass and it's not volatile, then we will be able to see it by the ELSD detector. The way this works is the material as it enters the instrumentation after it passes through the column, so that purified material passes out of the column. It's aerosolized in the nebulizer, and this causes a spray of fine droplets. These fine droplets dry as they move through the instrumentation, through what's called the drift tube. So that shell of solvent around these particles dries off, and then what you have are a series of dry particles that move through a detector, which in our case is a laser. As these particles move through that light path, they scatter light, which is collected by the photodiode. The more particles, the higher the signal strength, the larger the peak response. So why, once again, do I think this is important? Well, since it's almost a near universal detector, you can see compounds that you normally can't see with the UV. So non-chromophoric compounds. It also provides qualitative sample information. So things like ratios of amounts of materials in the system that you're purifying. So if you're purifying something and you have a large UV response, that could be because it's very chromophoric, but that may not represent a large part of the sample concentration. Material moving through the ELSD, the amount of particles in that particular product as it's being purified determines the signal strength. So that means larger peak responses means a larger concentration of that material in the sample. You can also, as I mentioned before, use chromophoric solvents, acetone, toluene, things like that, if that's going to help in your separation. You only need to look at samples that generate a peak. You don't have to collect all fractions and test all fractions, because if a peak is generated, that means there's something there. If there's no peak generated, then there should be no material in that particular area. So this minimizes the post-analysis fraction workup, decreases the amount of quantification steps such as HPLC runs, TLC plates, and the time, solvents, and materials involved. Let's look at an example of this in real life. So here's a minimized fraction workup, because what we're looking at here is we're checking peak fractions only. So on the top, we look at an ELSD response with UV. So this is our Reveleris X2. We're looking at collecting peaks only, and we generate 3 peaks. Peak 1, peak 2, peak 3. So now we test those 3 peaks, find our compound of interest, and we're done. On the bottom, we have a UV only system. So now here we see we have to collect everything. We only see 1 peak. So to be sure we haven't missed anything, we have to collect all the fractions. in this case 14, and we have to test all 14. So if each fraction takes X amount of minutes to work up, it takes 3 times that number for the collection that was done with the ELSD and UV together. It's 14 times that number on a system that used the UV only. So you only have to analyze the fractions that are visible or the fractions that are collected, and this saves, once again, a large time in sample workup and consumables. Sample purification. Flash, prep, both. Here we see some examples of what's available. Anyone that's done any type of chromatography has used a manual gravity column. Lots of sample can be loaded on these. They have poor resolution. They can take sometimes days to run. They're very inexpensive, but they don't have any detection systems. And so every sample that's generated off of this, depending on how it's done by different solvent or by time frames, each one has to be tested so that you can find your products of interest. And unfortunately, in many cases, this type of technique isn't highly reproducible. If we look at automated flash chromatography, so here we have our X2 system. Here we suddenly have a high capacity system also, but we have very good resolution. We have very fast sample purification. Instead of days, now we're looking at minutes. We can use inexpensive and disposable cartridges. Many phases are available. There's multiple detectors, UV and ELSD detection available. And the results on these automated systems are very reproducible. Finally, we have automated prep chromatography. While we don't have the same capacity, lower capacity, because we're using high resolution HPLC style prep columns, we don't have the same amount of material we can put on, but the resolution now has become very, very great. These are highly reproducible. And we also have any phases available. Any manufacturer's prep columns can be used on the system. Multiple detection options, UV and ELSD, and very, very fast sample purification. What's the difference between using flash or prep if we're saying that both do a good job at this? So if we look here at the top, we're going to do a comparison of assay done on a flash system and on a prep system using the same crude material. So we have a crude material that was run on HPLC just to get an idea of what the components were and where our compound of interest comes out, and have an idea of what type of solvent set and solvent system we'll need to use. On the bottom left, we see our flash separation. While we have relatively low resolution, we were able to find our compound of interest, and we had a relatively nice capacity, 10 grams in that sample run. If we look on the bottom right, we see a prep separation, very, very nice resolution, almost exactly the same as we see on the HPLC workup that we did. However, we have a much lower capacity because we're using a prep style metal column. When we look at the final results for LCMS flash purified fraction, LCMS of the prep purified fraction, we see that our compound of interest is purified in both cases. A little better purification on the prep side, a little worse on the flash side. However, the flash side could simply be rerun on either the flash system again or on the prep system as a polishing step, and result in a much, much higher purity of the final fraction. This particular slide, we're looking same thing at a comparison like this. We're looking at Citral and lemongrass. So we have our crude run, and then we have our purified material. And if we look at some of our results for purity and for recoveries, we find our results are pretty consistent. The flash model gives us 98%, and about 360 milligrams recovered. The prep system gives us about 99% purity with about 388 milligrams per gram recovered. And then if we look at a standard preparatory system that's available in the marketplace, we're looking at about the same. Here again, 96% purity and about 373 milligrams per gram recovered. So what we're looking at here is, a standard preparatory system can be a quite expensive investment along with the columns. What we can see here is that if we compare that to the flash prep system, we can either run flash chromatography and get very similar results. We can run prep chromatography and get almost exactly the same or better results as a standard preparatory system. And then we have the additional flexibility of having much larger capacities by going back and forth between the two systems, giving us much more flexibility. Let's look at some examples of increases in productivity and where these increases are coming from. So if we look at this simple synthesis that you might see at any type of med chemist or pharmaceutical chemist doing, you're taking some reactants, you're running some type of reaction with them, you're producing a product of interest and some residuals, which can be unreacted material, it could be other products, secondary products that you're not interested in, and so on and so forth. So we run this reaction, and then we find that we're going to follow the flash purification process using a UV only and a Reveleris ELSD UV system. And we find that if we look back at all those steps we talked about in the process of purification, sample workup, sample screening, flash separation, post-sample workup, post-sample analytical testing for identification. We're looking at all those steps. Now we find that we have large savings in time. So if we give a time frame to some of these that was done in this previous lidocaine purification process, we see that by using the UV-only system, it's taking us much more time. In many cases, it's taking us longer to set things up. It's taken longer to set up a good method to make sure we have everything purified. It's taking us longer because we have to collect everything to make sure that we haven't missed anything. And it's taking us longer to test all these extra samples to see where our actual purity is and if we can account for all the fractions that were produced. On the flash system with the ELSD and UV, our Reveleris system, we find that a lot of these steps have been decreased in the amount of time they take because we're now limiting our testing in most cases to only the fractions that are collected, not all fractions. So only peaks collected need to be tested. And this ends up saving us about half the time that it would take us to do the same analysis, the same assay and analysis using a UV-only system. Let's talk about some other ways we can use some of the features on the flash system to increase productivity and achieve optimal results. The Reveleris flash system has a Navigator software feature that allows for a rapid method development to do your purification. These purifications that are suggested by the instrument can improve the quality of the separation, they can increase the speed of the separation, they can decrease the solvent consumption, or they can increase the resolution between peaks. The initial input that's required to use this software feature is either an HPLC run or the TLC RF values and conditions that were used. This can optimize the gradient starting conditions for the flash purification. If the results aren't well enough separated on the paradigm that's inputted into the instrument, the Navigator feature will warn you that that separation probably will not work as efficiently as you'd like it to, and will suggest using different solvent sets. And later on, we'll also see that, in some cases, two solvents simply isn't the most efficient way to do an extraction, especially for people doing natural products, where they may want to go from a completely aqueous to a completely organic system to be able to not only remove the water-soluble components in their sample mixture or their crude mix, but they may also want to be able to purify and extract out the organic or the lipid-like components. Let's look at an example of how this would work. So here's a reaction that's run. We have reactants, we have a reaction mixture, and then we have a final product, and then obviously we have some unreacted materials left over. So this product of interest for us will need to be purified. So if we open the Navigator software feature, we're going to see that we have some TLC plates on there. We have some solvent choices. What we're going to do is input our data. So we're going to put in what solvents we used. We're going to put in our RF values for 2 different concentrations of solvent, a low and a high. We're going to tell it what size cartridge we want to use, the flow rate, if we want to purify things quickly and save solvent, or if we want to purify things for a higher resolution. And then if we want to continue to 100% to flush out the cartridge at the end of the run. Let's look at what these choices suggest. So if we choose purity, you'll notice that we're starting at 0% B solvent, and we're running a shallow gradient, perfect for the HPLC style pumps that we have on this system, and to generate a high purity run. On the speed side, we can notice here that we're starting at a higher percentage of B, and that we have a little less shallow gradient. And then what we're going to find here is that this run is going to end up being a lot quicker for us. If we look at the results that these 2 particular assays generate, on the left side here we have the purity run. On the purity run, we can see that we start at 0% B. Our material is more retained, and we also have a nice separation where we have multiple peaks at about between 4 and 6 minutes. If we look at the speed run, on the other hand, now we're looking at our material being done by 3 minutes. So if compounds are collected within the time, by the time frame, you can always raise the percentage of your B solvent to 100%, flush out the cartridge if need be. If the cartridge is not going to be used again, the assay could simply stop there. So now we're looking at a minimum of 6 minutes or so on the purity one, and looking at a minimum of 3 minutes on the speed. So once again, we're saving about half the time, half the solvent. What happens for our 2 results from these? Results ended up being very good. We're absent of the starting materials. We have an NMR trace and a GCMS trace, and we find that we have near 100% purity, and that was done in this predicted gradient profile from our initial starting conditions from our TLC plates. Let's look at another example. This is another synthesis where we're looking at a couple different reactants, a reaction mixture, and then the generation of a compound of interest, plus some byproducts. Here we've decided to use the LC transfer function in the Navigator. So instead of using the TLC plates, what we're doing here is we're going to take a little bit of that crude mixture, run it on an HPLC system, and then we're going to transfer some of that information here, mainly the retention time data, the starting point of the gradient, the ending point of the gradient, the percent change of the gradient, the dead volume of the system, what type of HPLC column was used, and the flow rate. And then we're going to input what size flash cartridge, what flow rate we want to use, and then we'll have it generate a separation for us. Here we can see it's generated a separation profile for us. So what's nice here is this gradient method, as soon as you accept it, it's automatically preloaded and pre-populated onto the run screen. The crude mixture was injected and the flash separation was completed. So now we can look at taking a very small amount, putting it on an HPLC system, running a scout run, and then moving to the flash system and putting a large amount of material on, and then doing a large purification. Here we can also see, if we look at this section here that's circled off, underneath that main peak, we also have a contaminant peak. So what this instrument was able to do, by having multiple UV detectors and the ELSD running at the same time, it was able to sense a secondary peak coming up underneath that large main peak. And what's of interest here is the instrument delineates that into a separate fraction. Now, if that's simply that compound poorly absorbing at 280, then once it's tested and you find out that your material is pure, that material can be put right back in. If you find that that's not part of your separation, then that material can be pulled out, and then you can decrease the amount of contaminant inside your peak and the number of reruns that you need to do to have a purified compound. So what we see here after our purification, we find our cuts here that were made, and what we find is, if we're looking at our fractions, we can see that we have a very nice set of GC, NMR, and TLC, showing us that we have a well-purified final product. Sometimes people doing natural products might need to do a much larger scale separation. Sometimes what you'll see is people will do step gradients and then change solvents and do step gradients, and then take those materials and then rerun them after they've been collected, and then rerun them, each of those steps. What we can do with our particular system, since we can use up to 4 different solvents in one run, we have a binary pump system, but we have a 4-valve manifold. So what we can actually do on the Reveleris flash system is run a set of solvents. So in this case of natural products, you could do, if you're doing some kind of screening or online extraction off of a solid load, you can run an aqueous extraction, say water methanol. You can change to a more intermediate, so you can say methanol, methylene chloride, or methanol ethyl acetate, and then you go to a fully organic, so you can go from ethyl acetate to hexane, all in one run. All you have to do is make sure all the solvents run to their next miscible point. So the next solvent has to be miscible with the prior solvent. So you don't have any miscibility issues that could damage your column, but this also allows you, if programmed correctly, to run a multiple solvent set in one run, saving time and increasing the efficiency of your extraction process. Here's an example, once again, of a natural product style assay. What we see here is very nice example of the ELSD working. We're generating a really nice ELSD peak on a compound, and then we find that we have a very high purity on this compound after we did the separation. See really low UV responses here, and that's mainly based on the fact that lots of natural products don't have a lot of chromophoric activity. And because of this, the ELSD can be of extra value to people doing natural products assays. Time scales. So this is really what it's all about. What's the difference between doing some of these? So on a manual system, you might be looking at a day to fractionate, a day to analyze the fractions, and then another day or two to run all the HPLC separations or quantitative analysis of all the fractions that were collected. Looking at our flash system, you could be looking at 15 minutes for your run, 2 or 3 hours for the analysis, because now we're only looking at collected peaks because we're using ELSD and UV together. Workup of those fractions, and now we're looking at half the time to have the same amount of results. So a huge increase in productivity can be found by using the right type of system for the right type of analysis. UV Corporation offers a nice chromatography portfolio. There's the prep system with the UV and ELSD, which is also has incorporated flash and prep. There's a flash-only UV ELSD system. There's also just a UV system for people that really only need UV. In some cases, that's sufficient. But in many cases, having the ELSD offers a lot of time savings, and in some cases can be indispensable, and such as an example would be natural products. We also offer some other modular systems, like our Sepacor system. This can be a quite extensive system with fraction collection and multiple detectors, or it can be a somewhat simple system with a pump and cartridge and pump controller. We also offer a standalone light scattering detector that can be used with your HPLC system to help corroborate the fractions that you analyze. So in other words, if you run UV ELSD on your flash system, it's nice if you have ELSD standalone on your analytical system so that you can also see the same style results. Then we offer, obviously, highly efficient cartridges of multiple different phase types and particle sizes that can be used with our flash systems to achieve very good purifications. So to summarize our talk, we talked about different systems for different laboratory needs and flexible systems that can suit multiple needs under the guise of one unit. We have a friendly software package that allows for quick use and people can get up to speed with it very quickly. The ELSD UV combination is a universal detection system that increases productivity by decreasing fraction analysis post-run. The Navigator function increases productivity by decreasing method development time. The Navigator function can help decrease solvent consumption and solvent waste disposal costs. And we found that we can use multiple solvents in a purification to increase extraction or to increase separation efficiencies. Questions are invited. I thank you for your time.